Okay, so if this works. So this is one of the tools I'm going to be testing, and it's wonderful. You just type in the name of your protein of interest. Some people might know about that one. And if you wait sufficiently long, so the JavaScript's working, is the Java working? That's the question. Yeah, so I'm going to jump ahead to this, just it's on the screen. So we spent 12 man-years developing this tool. The idea is to make protein structures easily accessible to everyone. Just type in the name of a protein up in the top left. We need an organism, so we default to human. You can choose other organisms. Currently, in the system, we have all of Swissprot. That's about 500,000 proteins, so we don't have every organism under the sun, unfortunately, not yet. Of course, we've calculated these data, and we could extend them for other organisms. That's sort of work in progress. I'm ready to go, which is cool. So, everything I'm going to talk about is a reaction to the reality of this, what we're all afraid of. A lot of people talk about big data and the data tsunami, but in the life sciences, and I think most of you will agree with me, that we're not overwhelmed with the volume of data. We're overwhelmed with the complexity of the data that we have to deal with. So I actually say big data is the little problem in the life sciences. And so how are we going to handle that? And that's how most people kind of respond. And so I'm here to bring some good news of hope, which is that we are built with an amazing data analysis system. Over half of the human brain is either directly or indirectly related to visual processing. So here's the hope. Can we learn to encode our complex, often very abstract data in ways that work with human visual perception? Then we can start to use half of our brain in analyzing things. And I'm not saying we shouldn't do statistics, Terry. Totally not saying that at all, but we should do the statistics and get better at visualizing statistical results in combination with experimental data in ways that really work well with our perception. For me, this was a big turning point because I started life as a mathematician physicist who always wanted to do biology. I was a nice card-carrying mathematician for 10, 15 years, doing respectable things like developing algorithms. But around 2000, I did something unrespectable and joined a startup company, Lyme Bioscience, which then went very large. We had our software was used by Celera to distribute the human genome. And at one point, I was actually responsible for the data visualization components across all of our software suite, which was challenging. And then I really realized that, statistics, bioinformatics algorithms, data processing, data storage, and so forth, all absolutely necessary. But at some point, a human being has to look at the data, and a light bulb has to go off inside of their head. And that's, I believe, the rate-limiting step of certainly the life sciences now. It's how good we are at creating a visualization in front of a scientist that can lead to an insight. And that's really what our team focuses on right now. And when I say data visualization, it's a catch-all. And the first one thing I definitely do not mean is imaging. I don't do imaging at all. You could do data visualization with imaging data, but most of the data that I'm going to be talking about are totally abstract and don't have a sort of a real space very often. And I really mean everything here. It's graphic design, user experience design, all kinds of data visualization tricks, visual analytics, interactive visual analysis, statistics, and then the entire gamut of bioinformatics tools. But the way we do them is we use all of that focused towards creating a screen that the user looks at and is a screen that can generate insight. And I'm going to be talking about a number of tools. Basically, the application of our approach to a range of different data, proteins, some stuff for DNA, and some resistance biology stuff. And then end up talking about some community initiatives that we've been running to help the broader community as bioinformaticians and life scientists get better at all of this, and then also briefly mention a project that we've been doing with Scale. So I came back from my excursion industry around 2007, and the first challenge that we took on with that sort of new approach that I had, trying to make things easily accessible and data actually usable by biologists, was this issue of gene names. So it sounds like a very easy thing. And we came up with a tool that actually makes this, can help biologists actually read literature. That was sort of the goal. And some of you may know this tool. Basically, it installs in your browser as a little button. We call it Reflect. And how often have you read a page and you see the name of some gene or protein and you don't recognize it at all, but when you cut and paste, go into Google, you realize that it's a synonym for a protein that you know quite well, but it's either old literature or it's in a slightly different field where the convention is to name that protein something else. And so Reflect is really an answer to that. So by pressing that button, within a few seconds, depending on your internet connection, the page disappears and is replaced by one where gene names and chemical names have been tagged. And when you then click on a tag, you get this little pop-up that appears just hovering over your tag. That gives you the idea was to create a concise visual summary of everything that a biochemist wanted to know about a protein just while you're reading the page, just right next to that tag. So just to drill into the protein pop-up a little bit more detail. You've actually got access to the complete amino acid sequence of that protein, domains from SMART, links out, of course, to lots of other places. And this was sort of the first foray into visualization, showing the best structure that we could choose at the time, the 5 or 6 most significant interaction partners in string, and then this is a visualization of sort of where in the cell the protein is believed to occur. And this is really just a placeholder. It's just it was an organism. In case it's from a different organism, you'll see a range of different images there. And I'll just ask you to keep those two places in mind, because later on I'll come back to them. I'll skip forward because we're running a bit late. There's an API behind the scenes, and you can tag all kinds of documents. I think it's 50 different document types, and use it programmatically if you want. It's became widely adopted. We have many thousands of documents being analyzed per day, and we're still working on, and this has been delayed for some time, the new version where we really have much nicer design and doing some interesting things like taking a whole page and now really looking at all of the genes in that page and coming up with some very interesting analyses like pathway analyses and so forth. And as mentioned, watch that space, because for about 7 years that's been a placeholder for some projects that I can now finally deliver on, and they're not quite back in Reflect yet, but coming soon. So this project I mentioned at the beginning, Aquaria, is about protein structures. This has been a labor of love. Actually, I really started this project back in line bioscience, where one of my original goals was to connect structures to genomic sequences. Structures, of course, are lagging behind genomic sequences in our ability to determine them, but Aquaria sort of is designed to bridge that gap. And it's really driven by the idea that I believe that the insight that can be gained by looking at structures of macromolecules can be enormous, and that's, of course, the prototypical example. We know there's a lot of structures out there. Actually, it's over 100,000 right now in the PDB, and which is a landmark in human understanding, but how often do you really go and look at them? And most biologists, the answer is very infrequently. But what percentage also does that 100,000 protein entries in the PDB? Some of them are redundant, actually. There's many cases of one protein being crystallized many different times. Can that be read? Yes. Okay. So if I was to visualize 500,000 sequences, let's say SwissProt, which are protein sequences we believe actually exist, are well described and so forth, where there's evidence on the transcript level, what percentage of those are actually covered by a PDB file? And that's only about a couple %. So pretty dismal, right? But on the other hand, we do know that's structures more conserved than sequence. So many sequences are very dissimilar but have the same structure. And so we can take the structures we know and map them onto sequences we don't know. For that, we need a method, and we looked at a meeting some people might know called CASP, Critical Assessment Structure Prediction. And in the 2009 meeting, which was published in 2010, there was one clear winner. It was a method called HH-Blitz that was better than all of these other methods at automated prediction of which structure should be best aligned onto this sequence. So we started working with HH-Blitz, and a couple of years later, this meeting runs every couple of years, they'd slipped to a number of places, so we're sort of in the middle of the project and had backed this HH-Blitz was no longer the winner. But then looking a little closer, we realized that all of the above methods actually use HH-Blitz as the seed and were hundreds of times slower. And given what we're going to do is very high throughput structural modeling, there was only one choice, HH-Blitz. I know there's statisticians in the audience, but I won't dwell on this just in the interest of time. We've spent a lot of time cross-validating the accuracy of the database. And basically what we can do now is, let's say for the 500,000 SwissProt sequences, we can tell you some structural information for 87% of those sequences. In fact, there's a lot of alignments here. This is essentially the largest systematic modeling of protein structures. And on average, for each protein, I can give you 85 structures that are linked significantly to some part of that structure. So for each sequence, you've got 85 structures to look at on average. That's a lot of information. So I think we know this, that structures match to different parts. This is a pretty an audience, I think, where a lot of this is a bit redundant. This is the P53 sequence, the one I mentioned before. So just sort of building up how a query looks, so we present sequence and structure in a very unified way to make it easy to navigate. By default, we choose coloring by secondary structure, and this is quite an important point of a query, we overlay that coloring with an annotation of every residue in the structure that's not equal to the sequence that you're really interested in. And this will become important in the example. I think I'll just jump to the example. Oh no, one more thing. So some people say we're sort of taking on or competing against the PDB. I don't really think so. PDB is designed for and by the structural biology community. If you look at that web page and see what's sort of most prominent, it's the title of the paper that describes the structure, the authors' names and institutes and so forth. That's appropriate if it's a resource for the storage of experimental results. What we're trying to do is something different, is try to put the sort of structural information first and foremost. And you saw how easy it was, just type in a protein name. And this is actually a really nice example. P53, very important protein. I don't need to describe what it does in this audience. And then which structure should we show? So the bottom part of this shows you all of the matching structures for P53, not just the structures that are identical in sequence, but everything that's detectably similar in sequence down to a certain threshold, really quite a low threshold. In this case, the lowest match is around 30% identity. That's fairly low. Then of those 130 structures, which one should we show you by default? And defaults are really important when you design software. We have a simple method. We choose the single structure that matches onto your full-length sequence with the largest number of identical residues aligned. So it's sort of maximum information content, if you like. That sounds like a good algorithm in general, and I think it is. In this case, I wouldn't say it goes wrong, but it points out something interesting. So it finds a structure that actually has it matches to a lot of the sequence of P53. So this region where my mouse is right now represents the entire full-length wild-type P53 sequence. And you see there's only structural information for part of it. And in particular, there's a region here where it's missing. So what the crystallographers have done is take two domains, fuse them together. There's no structural information for those two, they just fuse them. In addition, as I mentioned, that coloring scheme shows you see a lot of black marks. That's our way of saying something has been messed with. That's not the wild-type P53 sequence. There's actually 19 amino acid substitutions, many of them highly non-conserved, and they were added, as I read in the paper, to stabilize the structure. Now, it's fairly common in crystallography. Sometimes they have to do that. In this case, they've probably gone further than one would like. This is probably not a really good structure to look at if you want to look at real P53 structural information. We represent here that there's 87 structures that match in the central domain that binds to DNA. So we've got a nice way of taking those 87 structures and sort of breaking them down. So we break them down into what we call oligomeric state. And one of the really important states of P53 is when it binds as a tetramer to DNA. And we see also that, for example, do I see it on the screen? Yes, there's 44 PDB structures have a single molecule of P53 by itself. So you sort of know what you're going to get there. This might be a more interesting physiologically relevant state. So let's click on that. And just very quickly, sequence to structure navigation. So if I want to know where in the structure is the C-terminus, I can click, for example, down here, and that corresponding part gets highlighted. Whatever is currently selected, press return. That becomes the center of rotation. And double-click on the background, you zoom back to the entire thing. Notice that we've got a tetramer plus a DNA dimer. And if I click on any part of one structure, of one molecule, of one chain, that then becomes center of rotation and so forth. So it's a way of sort of taking a complex whole and choosing and isolating the focus on one thing while you can still see the rest. And vice versa. Click somewhere in the structure, the sequence view rearranges, and you see, okay, that's also towards the C-terminus, and you can move the residue around with cursors and so forth. But I think the really cool thing, and one of the reasons I created this tool, is this, which is to take sequence features, and they can be any aspect of the sequence. Because the thing is, essentially what a query has solved is, I've got the real full-length wild-type sequence, and I've got information about that, and I want to map it onto any of the structures that are available, and we've basically solved that mapping. And now we can take a group of features, for example, these are features indicating which parts of P53 bind to various other proteins, and now that becomes essentially a coloring scheme, and we can see which parts of P53 bind to various things. P53, really interesting. are these. So this is just the annotations from UniProt. Each is about 1,000 SNPs, essentially each associated with cancer, being derived from a tumor. The way we plot these, if there's a single residue, like this one here, 245, in one case there's a mutation from G to A, the same residue from G to C. So every time there's a different kind of amino acid substitution, we create a new feature lane. As a result, it just automatically shows you the two residues that have the largest number of identical or non-identical amino acid substitutions across these different tumors, essentially, arguably, the two most sensitive residues in p53. Let's choose the second most. Just move that around a bit. So now I'm looking at the 5 residues that are essentially the most sensitive residues, so many different amino acid substitutions that those residues cause cancer. And what is remarkable, we found, and we're writing a follow-up paper on this, is that there's actually a very nice clustering in 3D space, close to, but not exactly equal to, the DNA binding site. So that's something I can talk about later if people are interested. It's work on that. There's a nice paper about p53. But in the interest of time, let me show you what a query is really designed to do, which is to show you information about structures where there's no information available. Now let's choose GLUT4. Is anyone in the audience not know what GLUT4 is? Nope. No one does not know what it is. Okay. There's a couple of people. Okay. Terry, come on. So there's no structure of GLUT4 right now. It transports insulin. Glucose. But there is a structure of GLUT1 that came out about a year ago. And so you type in GLUT4, there's no structure. It automatically chooses essentially the best structure, which is GLUT1, shows you GLUT1 structure, as described here. That's the structure you're looking at. And the GLUT4 sequence is then superimposed onto that, and you can see where the structure and the sequence differ. So in other words, in this case, where GLUT4 and GLUT1 structure, sequences differ on the structure. So you can look at information about GLUT4, even though there's no actual structure for it. And just to show you, well, okay, I'll turn on the feature first. So there's a nice feature set here that marks which part of the protein is transmembrane or extracellular, extracellular or cytoplasmic. And to show you the value of being able to access all of the structures that are related to GLUT4, not just the best or one. In this case, what is it, 100 or so. Let's choose one of these guys. So this is a bacterial structure, quite distant, but still a sugar transporter. This is the same alignment. So we can see that the outside of the cell, which is up, it's kind of open, right? This is the outside open state where sugars from outside are believed to perfuse in. And then there are other structures that show an opposite state. And if I get the right one, it's going to be the xylose receptor 4JA4, which again is the same orientation, showing the inside open state. So that's the point. Being able to access all the different structures that are similar to the sequence you're interested in can give you insight into the operation of your protein. And the query just gives you that on the web page. And now I can skip through snuff. That was if nothing worked at all. Just some fun things we're doing. So a query is beautiful and easy to use. This certainly isn't. This is using a thing called parallel coordinates to visualize an entire ensemble of all related structures and navigate through this multidimensional data. I've a paper on that I can refer you to. This is kind of just for fun, as is this. You got to learn mouse controls, keyboard controls, hold down control, shift, zoom, and so forth. So we played around with things like using elite motion. It's very responsive to make that interactive. The software's all freely available if you want to play with it. Set up a demo somewhere. This is a nice idea. We're proposing it as a cover figure. Haven't got it on a cover yet, but that's essentially on the ground. That's UniProt, and each area represents a protein sized by the number of amino acids. And then grouped, as we see, by organism and so forth. So just the idea of representing or visualizing data sets, entire data sets, can be informative, and we're playing around with ideas like that as well. Something that's very new is we've invested as a community billions of dollars into determining these 100,000 structures, and I believe they contain a massive amount of information. We should take it more seriously. We're trying to work out how should we best show a protein structure just by default, because defaults, as I said, are really important. Most people never click off defaults. And in fact, if you think about it, most of the time you ever use a structure, you only use a flat two-dimensional representation. So let's try and get the best one. And the idea was to try and do that automatically. We're currently using principal components, so for each chain, we'll take the longest orientation of the molecule. I can't really see any objects here. iPhone, so sort of the longest axis, the second longest axis, and the thinnest axis is the one that's on the Z screen. So you can sort of see, get a better overview of the structure. That works reasonably well, but we've been testing to see how well. And so we're setting up a series of tests, taking 100 viewpoints around a structure. Systematically, we're going to be going through all of the main domains that are there and asking the crowd. There's a thing called Mechanical Turk by Amazon, where you can actually farm out a job like this to lots of people, which of these two views do you prefer, and aggregate the results into, I'll just jump to the data, where we see actually a very nice correlation between essentially people's preference and what we would predict based on a thing called viewpoint entropy. I know it's got an equation, but it's essentially a method that tries to take each segment and choose a view that shows you as much as possible every part of the secondary structure in a sort of balanced way, and that actually seems to correlate better with how at least our Mechanical Turk audience rate these things. So just ideas about how we can perhaps do better as a community in showing protein structures. And how am I going for time actually with all of that? Okay. Maybe I should slow down. This is not released. We sort of got a very positively worded response from the reviewers. Editor loves it, 2 reviewers love it, but we're going to do more work. I just sort of throw it in there. So we, of course, worked on a career because I love structures, but then we see there's always regions, or very often regions of a protein that we can't match. And so I had a student looking for projects say, well, why don't we look at those? Why don't we look at every region of a protein where we can't match it onto anything? So sort of we're a query of fails and all the modeling methods, actually. Yes, so if we take every sequence, sometimes we can match a sequence directly onto a PDB structure, okay? We call this a PDB domain. Sometimes we can't, but using a query, we can match part of the sequence onto a structure essentially using homology, right? Down to a certain threshold, wherever you set that, but at some point you have to set it. We call that a gray domain, let's say. And then there's everything else. Let's call them dark regions. And then there's a case that maybe the entire sequence doesn't match anything that's in the PDB. We will call it a dark protein. And this is, wow, I didn't realize it built up like this. This is how it looks for domains of life. Surprisingly large amount of archaea bacteria we can actually predict, even though this is what we actually have measured. This is what's in the PDB for those guys, like tiny percentage, but most of their proteome, so this is the entire proteome. I put all proteins in each domain, essentially end to end, and then sort of broken down to those categories. So essentially we understand like 90-something% of archaea and bacteria proteome in terms of we'd match it onto a structure and we then of course have some idea about function. But for eukaryotes and for viruses, about half of our proteomes are unknown, and about half of those of the unknown proteome are in a group called the dark proteins. So they're entire proteins where there's no reliable match or detectable match onto anything that we've seen structurally. So they're interesting proteins, and we have been characterizing those. The interesting thing is the first thing you'd think of as a structural biologist, that's disordered. And, well, that accounts for some of them. And then you'd think, well, they're transmembrane, that accounts for some of them. And then there's about half of the dark proteins that are neither of those, and we don't really know what they are. But I think I threw this next one in for Terry, just to say that there's very interesting differences. Oh, boy. Remember I said I haven't actually seen the slide deck run through before, so never mind. There's distinct differences in amino acid composition. Still clicking here. And the strongest signal that we see, surprisingly, is not that they're transmembrane, although we see a strong overrepresentation of dark proteins in the endoplasmic reticulum, but actually the strongest signal is extracellular and also correlates with things like post-translational modifications, in particular disulfide bonds and so forth. Actually, I came kind of from NMR, and I talked to some of my NMR guys, and they said, kind of course, they knew that secreted proteins are tough, but I don't think that awareness is really in the community. So the paper hopefully is going to help focus attention on sort of rolling back the dark proteome. And just to briefly mention, and this is while on the whole topic of proteins again, these two regions in Reflect. So we have 2 new tools in collaboration with Eliza's lab in Copenhagen. I don't know which example to choose here, but one's called compartments, and that basically, all of these, we sort of do the same thing. We get all the data that's out there and try and aggregate it together, various confidence thresholds and so forth, try to make it easily accessible, and then visualize it so we can see where in or in the cell pp3 occurs, or and roll over and find the evidence that it's in the nucleus based on these different things, and drill down things, and everything's got APIs, and if you want to access it programmatically. More recently, we did the same for tissue. So which tissue in the body of proteins expressed, pretty important. We looked at intersecting all the major databases that are out there. We thought the paper was important. A whole bunch of journalists didn't think it was important. It finally has just been published or accepted. I think it's available online, I believe. Anyway, certainly available, the resource. I don't know, anyone can yell out a protein name if you want, but that does a nice job of this, and it's been excellent having a graphic designer in the group who can really make this beautiful picture showing where these proteins occur. So this is sort of proteins, a bunch of tools there to try out tissues and compartments. And I think at least some people at WEHI know about this tool that we're currently in the process of developing. Actually, we just sent it off last weekend. It was part of my weekend to Nature Methods. So the vision is, this is of course Drew Berry insert, is that couldn't we have that for chromosomes? I mean, we have for query, couldn't we just sort of easily explore the structure of different chromosomes under different conditions and so forth? Wouldn't that be great? Of course, complex chromosomes are too large, too irregular, and so forth, and they fall on the mesoscale often, so we just don't have good experimental handle on it. But one has been developed, so some of you might have heard of Hi-C, that essentially puts crosslinks. I mean, formaldehyde is one of the commonly used ones, but catches together two strands of DNA, either on the same chromosome or distal chromosomes, in one particular phase of the cell cycle, and then by a process of that I fully don't understand because I never really did molecular biology, but we are able to determine a contact, basically. Just skip over this. Time is running a little short. So basically, these data sets are being generated, and this is I'll forget about this experimental limitations of these. These data are being generated. So I'm just going to skip and narrate, so don't pay too much attention as I'm jumping forward here. There's a lot of problems with these data, high false positive rates. There are some solutions to that or mitigation strategies that are being developed, and it's not here, of course. So essentially, each experiment now has millions of these contact points, but you sort of aggregate them and chunk them into blocks. And that's just one experiment. Actually, new experiments are more than millions. There's certainly billions of total contact data out there. So there's a couple of tools that can visualize these data, but nothing really interactive, nothing really sort of useful. So we've built this thing called Rondo, and we hope that Nature Methods agrees with us that it's going to be a tool that makes these data sort of really much more accessible. So we're looking at, this is sort of like with Google Maps. If you look at Google Maps for Australia, you don't see much detail. So there's one data set we're looking at here. It's for human embryonic stem cells. There's millions of contacts between the chromosomes being represented, but we're aggregating them down to sort of show sort of overall patterns. You can hover over one part, and that will sort of highlight that part in particular. You can click on a chromosome, and that now will, if everything works, it looks like it's working, we'll zoom into chromosome 7. It shows you now cis connections or connections within the chromosome. Remember that I said we chunk things down, so these are sort of chunks, and we're looking at all the contact from anywhere in here to anywhere in there. So we see there's a lot of contacts between those 2 points. And if I want more detail, just hover over them. Oh, where was I? Oh, anyway, it doesn't matter. Just hover over a region that you're interested in, and that region will become expanded. You can see more detail exactly where those contacts are. What you're sort of seeing off to the sides are, we're using sort of a rainbow map, which is a classically bad thing to do, to show contacts from that part of the chromosome to other chromosomes, colored. I mean, you can't see necessarily that particular shade of blue is chromosome, whatever it is, 10 or 20, but you can see that, there's sort of very few connections to other chromosomes in that region and more in that region and so forth. So you get an insight. You have to sort of hover over the connections themselves to really reveal these details. I don't actually have the perfect example, unfortunately. It's the wrong data set. And we just wrote the manuscript tonight. But you can type in a gene name. You can type in actually a pair of gene names. And you can look at, are there connections between those two gene names in that experiment? And that experiment is based on a particular cell type, and so forth and so forth. You have to go and read the data. You can set the different data sets, and you can compare data sets. You can actually have a thing where you can compare two data sets. And of course, we're looking at for things like cancer versus non-cancer, or comparing epigenomic states of two cells. And you'll notice that as you zoom down, the representation of the chromosome gets progressively enhanced, kind of like, as I mentioned, Google Maps, that you get sort of more detail as you zoom down, eventually seeing genes, epigenetic marks, and you can turn on a lot more marks. I mean, it's all quite extendable. That's going to be Rondo, coming real soon, and timing. Oh, okay, okay. This was just published, and this was a really nice collaboration with David James's group, formerly at Garvan, who I guess many people know was a foremost expert in obesity, diabetes research, energy metabolism. Basically, they had a data set, a landmark data set, the most extensive, or I've forgotten exactly the correct way of describing it, but a very large mapping of phospho events in fat cells in response to insulin and glucose stimulation. And they found something like 8,000 phospho events occurring. They measured them at various time points. They'd aggregated them together so you could see that a whole group go from low phosphorylation to high phosphorylation, stay there. Some sort of do the opposite, some go up and down. And so that was, and then they sort of clustered them into groups. Then they used this color coding to indicate sort of a grouping. And then they did what is very typical in biology, try and draw a sort of a map or a representation of a pathway. And it's a little difficult to work out what's going on. So we thought this would be an interesting challenge to apply our approaches, to see if we could take these data, a very important data set, and try and make them more lucid and rearrange these data in a way that can actually generate more insight. So we were inspired by this, by what in data visualization is okay. Maybe hopefully it's just that figure. And I can move on to the next one. That's a very famous figure. It shows the march of Napoleon's army from Paris to Warsaw and back, and the width is the number of soldiers remaining. It's regarded as one of the best visualizations. It's got multiple dimensions of information, their temperature, elevation, river crossings, and so forth. We took the idea of a journey, and we thought, well, this instant response data set, time series data of this kind we could potentially lay it out, sort of wrapping time around the cell. And so we took these data and did that. This was the first version. We have an interactive version where you can sort of hover over a given phosphorylation event and the corresponding entry in the heat map gets displayed, which is the best way because you can really see this is kind of an approximations we placed each phosphorylation event by the time of half maximum, the time point at which you went from sort of the lower state to the higher state estimate when you went through 50% phosphorylation, and that's how we use to place the phosphorylation events. Every time you draw a graph, there's always these hub nodes, right? These guys that get in the way and have got lots and lots of connections. So we pull them out with time. So the phosphorylation events or partners, particularly the kinases, have multiple events at multiple times, and they become a track. Whereas the things that have essentially one event at only one time are just single nodes. And the arrows connect kinases to their substrates. And the great thing is what we call in data visualization spatial reasoning. You don't have to, it's organized in order of time, and of course in order of subcellular context. So the layout frees your mind up to, you don't have to remember or track back the colors and so forth. It's just the layout actually helps you in interpreting these data. Indeed it has. Just last month we got I think I'll have a link here somewhere. It's in Cell as one of the so-called snapshots. There's two links to it. Actually, they're even linking to our page, which is slightly nicer. And there's a lot of information here. So if you hover over, for example, an individual a phosphorylation event, there's text describing exactly what that event does. You get the kinase and the substrate tracks highlighted if there are tracks. A really nice subtle animation effect going on there. I don't know if you can see, but showing the switching of the cell from the initial secreting of fatty acids, that gets turned off around here. Glucose gets uptaken, and under some circumstances, lactate is secreted. So it's just a clear way of taking this really important data set, making it really visible and usable to the community. And this is a great example of the kind of thing we like to do. But it's great that we can do this. Why can't other bioinformaticians do this? And why couldn't I do that in the first 10 years of my career? And that was a question that a friend of mine myself thought, well, we've got to raise our game in bioinformatics and in biology in general. Our data sets are too complex, and it's no longer sufficient for us to be mediocre with data visualization. It's something you have to take seriously. So we created this thing called VisBee, Visualizing Biological Data. It's an international conference series. We just had a 6th meeting. They've been really successful. We get into one room, designers, data visualization experts from computer science, human experience designers, with bioinformaticians, and we try to get the best in the world. We had UCSC, for example, the tools that you're actually using. We try to get the people who make those tools together with the experts in data visualization, side by side, hoping like consenting adults, they'll work out what to do. And it is having a big impact. We launched the first meeting with a special issue of Nature Methods. We were very fortunate to engage the editors at the time, and the response of the community was astounding to that special issue. So it shows us that there is a crying need in biology for clarity, simplicity, better data visualization, and this really had a big impact and is continuing to do so. So there's our website. We put a lot of effort into doing that, making the results and outcomes of the meeting accessible to you. So there's posters, there's videos, almost all talks. Welcome to come to the next meeting next year. And finally, that meeting brought me into contact with people like Gail McGill and Drew Berry. And particularly Drew Berry, when I came back to Australia 3 years ago, we'd actually had this idea of VisBee as a conference for us, for biologists, for bioinformaticians, to learn how to get more insight into our data. But there's always people like Gail and Drew around who are great at that, but they're also superlative at not only just using visualization to discover new insights, but to communicate those insights to a broader audience. And we thought, well, could we have some meetings or somehow have something related to that? Sort of, we called it VisBee Plus. And so we convinced the Australian government, and Drew was really instrumental in helping us do that, to fund the 3 new animators to be trained up. So we're essentially outsourcing Drew. And because there's only one Drew Berry, but there's many stories to be told. And so we have released now 5 stories. We had actually just the 6th one just released on the weekend, still not up on the website. So there's a CSIRO, WEHI, and Garvan Institute. Each has an animator that's been trained by Drew. And 2 of the animations actually were selected in the Visualization Challenge run by Science Magazine and National Science Foundation in the top 10, which was awesome. And also, just last month, we got the whole project was awarded an interesting grant, the New South Wales Emerging Creative Talent Award, which put us in a room with some very interesting people who were definitely the only scientists there. So I was very, very proud of that. So, to sum up, what do I want to say about this again? Yeah, I have kind of a message that I want to give to students, group leaders, institute heads, and particularly funding bodies. You know, I mean, we have this data coming, and we're generating it, most of us. And what good is it going to do if we just continue to generate increasingly complex data that's already, in many cases, overwhelming, without a plan as to how we're going to organize that data into forms that can make it actually interpretable and able to deliver insight? What's the point of that? That if we do that, we're contributing to the confusion of the world, not the clarity. And that's, I came here and became a scientist to do the latter. And if we can learn to visualize data correctly, and if you can see through the things, hopefully we can actually learn to ride the wave, as it were. So if you're interested, there's another meeting we have called BioVis, more computer science oriented. It's coming up next month. There's a meeting we're running down in Hobart. This is sort of less biology, but still some very interesting work if you're interested in following up on that. Next VisBee meeting's back every year. We alternate every year from EMBL in Heidelberg to the Broad Institute in Cambridge, Massachusetts. And we just had a data visualization masterclass where we got, Gail was teaching us how to storytell. We had a world expert in computer science data visualization and an excellent teacher. It was really successful, and if anyone's interested in organizing another one of these things, get in touch. We got the sort of thing together, and I think I'll need a few more weeks to think about it. But I really have to thank Drew, whose support has been enormous in launching the VisBee Plus project, which I haven't really touched on because I know that everyone here has sort of seen most of Drew's animations and Maya's and so forth. So, so that's it. These are the tools. Definitely want to thank the members of my lab, and thank you for your attention.
So any questions for Sean? So So, Sean, so that your website is the best place to see all these things, and is there demos, or how do you take on and learn this stuff, apart from seeing you?
Well, of course, we try to design most tools to be easy enough that you don't really have to learn. We have a YouTube channel, Untitled Lab, where for some of the tools at least, we've given sort of creative video tutorials. We've been talking to Gail, whose new Clarify site that was very interesting as a way to deliver teaching information like that. And I'd also be interested in running other trainings if there's other opportunities. But yeah, one way is to try to make them simple enough that we don't kind of really need training. So it's a goal at least.
The metabolome and the microbiome are the big tsunamis that are on. What are your thoughts as to how you might be able to organise that data to make it more digestible?
No pun intended, I assume. So we started looking at that. So for example, the Rondo view, we built it to show connectivity, 3D connectivity, but in reality, there's about 1,000 data sets out there, high-C data sets, but there's a lot more data sets on for example, microbiomes and other kinds of microbes. And we could definitely, we're actually sort of thinking about how we could repurpose the tool to show some microbial data. So we just started to fire in that direction. I mean, I think for that, we really need a research grant, and we don't currently have one in that space. So if you want to talk to us about that, or Liz. I just think, well, I'm running the VisBee thing, and we get every year people in microbiomes and metabolomes coming, and so you can check on VisBee. We have at least one speaker in that field every year, so you can go back to the history of the videos and see what's there. I'm aware it's a field that's really struggling. It's tempting to me because that's exactly what I like. I like going into a field where there's this data which just no one's got any idea how to visualize that. That's kind of fun for us. I just haven't been fun to do that up until now. So, but I think the fact that what we're doing with VisBee is giving everyone the resources, providing people access to resources that can be used and are being used in that field. So, more I can't say.
When I used to go to church as a kid, I always thought the priest or the minister was speaking directly to me when he was talking about the sins of some broad category of people. And I'm feeling that with you. And I want
I was, actually.
I want to put a pitch for another sort of graphics, neither yours nor the one that preceded you, where you're actually visualising to address a very limited number of questions, maybe only one, maybe a small number, so that you display your data in a particular way, and if it looks good, you move on, and if something unusual happens, you're led down a different path. And I spend a lot of my time trying to dream up visualizations like that, that either reassure me or draw my attention to. You guys are presenting what I call generic visualizations, and it's not entirely clear to me what you're supposed to learn, except, wow, this is good, and look, yeah, it's sort of making it accessible and easy. But from the point of view of like, a hard-nosed guy like me, there's no take-home message usually, except, gee, that's beautiful, or informative, or something. Now, is that spectrum of sort of very specific visualizations addressing a question versus what I'm maybe insultingly describing as generic visualizations, which are pretty and in some sense appealing to journal editors or covers of journals or textbook writers? Is that a recognised dimension in your business? And would you give me a little bit of credit for being at the other end?
Well, there's a recognised spectrum of, as Gail was saying, explaining versus exploratory, right? And I think you're really towards the exploratory, and you would very, very focused problem, and you want to see the visualization just has the information you need and enables you to explore just those dimensions. And we certainly do things like that too. It's true, we're generally building more generic tools. And I did focus, remember in Aquaria, I could go back to it, but I showed you these are genomic sequence variants that cause cancer, and we see that they cluster. That's an insight, a specific insight, one particular domain of a protein, its interaction surface with other proteins where we're really seeing some insight. That's an important visualization. That's not just generic, that's not just pretty pictures, that's insight. The Rondo view, I mean, I didn't bring it in because it's very new, but we are going down into different regions, and we're comparing stem cells versus differentiated cells in particular regions where we know, like the Hox cluster and so forth, and we're seeing conformational changes that you sort of have to learn how to read them in there. And I just just glossed over it a little bit, because I'm just basically trying to give you sort of the breadth rather than deal too much in the depth. So I think I would refer you to the P53 example, the one I did show you. Happy to show you the one on with Hox and so forth for Rondo to convince you that such visualizations are there. And I could also show you lots of other really tailored and custom visualizations that I haven't shown you at all, because I think they're kind of answering the kind of questions you like. In fact, I've got graphs that I'd really love to show you, but maybe can I even bring it up? I'll see while I'm answering the next questions. Is there another question then?
Yes?
One of the things that there's a lot of work that's done on visualizations of this type, but so often there has been very little actual proper evaluation of the effectiveness of it. And in a way, it comes back to Terry's question there, because a visualization can often be there to assist in carrying out a task, right?
Yep.
Now, when people have actually tried to evaluate against a task for a lot of visualizations, they've failed to get an effect, right? Particularly, for instance, 3D, working through 3D visualization, different types of manipulations. Colin Ware did a lot back in 20 years ago.
Mm-hmm.
Have you made any effort to look at the question of evaluation of the effectiveness of visualizations?
Yeah, so we did, for example, we did a short user study with the leap motion to see if that was actually more effective and easier for people to just to manipulate a protein structure rather than having to learn sort of keyboard controls. I mentioned briefly that work we're doing on evaluating the best viewpoint, and we're doing another study. So, and I think a very more specific one. So the marking that we're using to show quality in a query. So you come to the query structure, and if the structure you're looking at is exactly corresponding to the sequence you want, it's going to look beautiful. It's going to look really nice saturated colors. It's clear. It's going to look something like this, right? And we're communicating that's 100% sequence identity between the sequence you're interested in and the structure you're looking at. This is, what is it, 27%. So we're now communicating quality with visualization. And we're, for example, actually we just submitted yesterday a paper on this, on evaluating the effectiveness of that kind of communication of data quality, which I think is critical. So is that an answer to your question, or are you looking for a more generic kind of test?
I think what you're saying is that you are getting actual task-oriented feedback.
Yeah. Right, so there's a thing called Mechanical Turk I mentioned. So we take bunches of images like this, we present them to hundreds and hundreds of people at different quality levels, and we ask them to answer certain tasks. And the result is kind of the statistics I showed you before, but we're able to work out how effectively is data quality being communicated visually. Really, and across hundreds of people, not just the 3 people you happen to be in contact with and what they thought about it. So we're definitely doing that, and I think this is one of the key things. This is a fairly sort of an original encoding of data quality in a visual context. And it's very important in query because a lot of people are going to come to a query won't necessarily realize that that's a model. So pre-attentively, before you thought about it, you can sort of see something's up, right? And you can go and find out, at the very top there it says 27% identical, that the structure you're looking at is quite different to the sequence you're looking at. But we want to communicate that pre-attentively, and we're testing exactly that. So I think that's one example where we're doing that. And the platform, Mechanical Turk, that Amazon offers to access this community of people to run these tests at real statistical scale is great. We've actually built a couple of tools we're probably going to publish as well to help other people do similar tests.
Okay, we're out of time. So we have to thank Sean Downey.